All right, this video will cover gluconeogenesis. So gluconeogenesis, as you maybe would have guessed, is making glucose from scratch, right? So neo is new, genesis is making stuff. So gluconeogenesis is making glucose from scratch. You already know that this happens in plants and other photosynthetic organisms, and those organisms use a process called the Calvin cycle. In order to fix carbon dioxide into glucose. We will learn about that later. But today, we are learning about gluconeogenesis, which actually takes pyruvate and runs it back to glucose. So gluconeogenesis is backwards glycolysis. Okay? All different types of cells can do gluconeogenesis, including photosynthetic organisms. We do it, bacteria do it, everybody does it. So gluconeogenesis, as I just said, is making glucose from pyruvate, and so it requires NADH to donate the electrons needed to create the new bonds, and is essentially backwards glycolysis. In fact, there are, so this is, let me see if I can zoom out a little bit more than that. Okay, so this way is glycolysis. This way is gluconeogenesis. There are, of each of these steps, however many steps there are 4 that are irreversible steps, okay? So the irreversible steps are the steps that will be either used for glycolysis or used for gluconeogenesis. The other steps are reversible steps, and they can be used for either process. And so gluconeogenesis and glycolysis actually share a lot of the same enzymes. We will learn none of those. What we will learn are 2 of the irreversible or dedicated gluconeogenesis enzymes. And so the first one we'll learn is pyruvate carboxylase, and the second one that we will learn is fructose bisphosphatase. So we are going to learn the names of these enzymes, what they do, and how they are regulated by the cell. Okay. All right. So here you are doing you're doing glycolysis. Down this way is glycolysis. Okay. So glycolysis takes us from glucose to pyruvate. As you guys know, the next step, pyruvate oxidation, takes us to acetyl coenzyme A, which you cannot see on there. I will just scooch this up some. And then acetyl-CoA will go into the Krebs cycle. All right, so there can be a situation, which I have right here in this other part of the slide. So there can be a situation where here's the acetyl coenzyme A, and in the first step of the Krebs cycle, it has to bind with the Krebs cycle intermediate oxaloacetate. And so if glycolysis is happening faster than the Krebs cycle can happen, then you will have too much acetyl-CoA for the oxaloacetate. The other way that this can happen is, if you guys remember from the eating other things besides glucose video, fatty acids are broken down by beta oxidation into 2-carbon sections and enter the Krebs cycle as acetyl-CoA. So if a bacterium is digesting lipids, it could also be making more acetyl-CoA per unit time than can be accommodated by the Krebs cycle due to the limitation of oxaloacetate. And so the cell needs a way to harness this acetyl-CoA so that it's not just wasted, even though the cell can't use it right then. And so for that, we have the enzyme pyruvate carboxylase. And so pyruvate carboxylase is activated by excess acetyl coenzyme A. And so, to be frank with you, I've just realized that I have no idea how we reverse pyruvate oxidation. We're just going to say that we don't. And so pyruvate carboxylase is activated by excess acetyl coenzyme A. Somehow, that excess acetyl coenzyme A makes it up to pyruvate, and then pyruvate carboxylase changes that pyruvate into oxaloacetate, which is this Krebs cycle intermediate that we are currently short of. And so the action of pyruvate carboxylase gives the cell the option of whether to do more of the Krebs cycle or to continue up toward gluconeogenesis. So hopefully that makes sense to you. Activated by excess acetyl coenzyme A, allows for more / quicker Krebs cycle, or the cell could keep going up this pathway and go to gluconeogenesis. Okay. Pyruvate carboxylase. Let me write over here. And it does pyruvate to, can you guys see this? Yes. Okay, so pyruvate carboxylase does pyruvate 2 oxaloacetate. It is activated by excess acetyl coenzyme A, indicating a slowdown or stoppage of the Krebs cycle. And so by providing more oxaloacetate, the cell should be able to get the Krebs cycle running again. If it would like to do that, if it would not like to do that, then the cell can continue on to glucose. There are lots and lots of other different signals happening in the cell, which we will not learn. This is just like a pretty straightforward one, so I thought we would learn it. Okay. The other one, which is relatively straightforward and we are going to learn, is in this extremely pizzazzy but hard to read orange box, and it is called fructose bisphosphatase. Okay, so fructose bisphosphatase is an irreversible gluconeogenesis enzyme that functions pretty close to when we get back to glucose. And so it takes fructose 1,6 bisphosphate, which is a 6-carbon sugar with 2 phosphate groups on it, and turns it into fructose 6-phosphate, which is a 6-carbon sugar with 1 phosphate. And so the super nifty thing about fructose bisphosphatase is that it is inhibited by AMP. Okay, so pretend like don't pretend like. So you have, all of the adenine something phosphates in the cell. Okay, right? So like the more of this is ATP, the less is going to be ADP and AMP, right? Because there's like the total amount, and if one section is bigger, then the other sections are smaller. This is like normal fractions. Right. So the cell is fine if it has lots of ATP. Woohoo! The cell is in a pickle if the amount of ATP is very small, but the amount of AMP, adenine monophosphate, is giant. Adenine monophosphate does not carry a ton of energy because most of the energy is stored kind of in between the phosphates. This is where my lack of biochem comes shining through. In any case, having multiple phosphates makes the compound be able to carry more energy than having one, due to the interaction of the large and negative phosphate groups. So AMP is very energetically poor compared to ADP and AMP. So if the cell has lots of AMP, right, if it has lots of AMP, then it is weak and ineffective. And so it really needs to make ATP. Okay, everybody got that? If you have lots of AMP, what you need is not to make more sugar. What you need is to break down any sugar that you have to make ATP. And so fructose bisphosphatase, right here, is inhibited by AMP. And so the more AMP you have, the less, so more AMP makes this step happen less. And so the more AMP you have, the more this happens. And so then we can go into the Krebs cycle and then on to electron transport and oxidative phosphorylation. So hopefully that makes sense to you guys. That is the 2 regulatory steps of gluconeogenesis that we are going to learn.